Hello, we are the Grimes Lab. We study the bacterial cell wall known as peptidoglycan and how it interacts with the human innate immune system. We are a very interdisciplinary group that utilizes areas of organic chemistry, biochemistry, chemical biology, and immunology. Major techniques prevalent in our research include carbohydrate and peptide synthesis, chemoenzymatic synthesis, bacterial engineering, protein purification, and immunoassays. Our offices and labs are located on the first floor of Brown Laboratory. We have two lab areas that are identical in floor plan. The general layout of the work areas are one organic hood at each end and two in the middle with a biochem bench behind each hood. This organic biochem pairing allows our members to be exposed to others working on different projects and areas, thus encouraging everyone to learn about their colleagues' work and explore new techniques from vastly different fields. Across the hallway, we have two group offices with plenty of space for our members to read scientific literature, analyze results, and communicate ideas to other members. Our lab is well equipped for conducting our syntheses. We synthesize various compounds such as carbohydrates, peptides, fluorescent molecules, and chemical probes. We also have two in-house HPLCs for purifying our newly synthesized peptidoglycan fragments. Across the hallway, we have our own Waters Acuity UPLC mass spec to monitor our reaction progress and identify our samples.
For a biochemist, we have small and large temperature-controlled shaking incubators for culturing bacterial samples. Here's Liz growing up large cultures to extract and purify protein from. We also recently installed a dirty culture hood for working with our BSL level 2 pathogenic bacteria. Here's Kimi inoculating her Pseudomonas aeruginosa samples. Upstairs, we share a tissue culture room for growing mammalian and insect cells, as well as preparing samples for in vivo assays. This is Annie changing media for her Higgs cells. Many of us also purify recombinant proteins for subsequent in vitro assays to study protein structures, functions, and interactions. We have recently updated our SPR system for analyzing protein binding affinities. Here, Liz is conducting an experiment to see how tightly a protein receptor binds various synthetic peptidoglycan fragments. We also installed a microarray for one of our projects led by a senior member in the lab to study the role of peptidoglycan fragments in adaptive immune response generation.
Our lab has a nice, welcoming, and collaborative environment. Many projects integrate both aspects of biochemistry and synthetic chemistry. Often, we see synthetic chemists working and learning from a biochemist, and vice versa. We like to be very translational so each member will become knowledgeable in multiple fields and techniques.
What is the point of research if you do not enjoy it?
As a lab, we create a new t-shirt design every year to represent our lab in a fun way. Before the pandemic, we would go on lab retreats, have picnics at Lum's Pond and White Clay State Park, and celebrate individual members' accomplishments with ice cream from Beauty Creamery or Cafe Gelato.